Yeah, I'd like to first thank the organizing committee for inviting me to attend this program so that I could share with you some of the recent findings of our group and also in the current literature regarding the potential role of stem cells in cancer of HPBs. So when we talk about cancer stem cells, what do we actually mean? Well, basically, in the past, cancer cells were all considered a homogeneous population, so all cells would have the capability to proliferate and form tumors. However, over the past 10 years, emerging evidence seemed to suggest that the ability to initiate and sustain tumor growth is more or less dependent on a small pathological pool of cells, which scientists have termed cancer stem cells. So they're termed cancer stem cells because, similar to normal stem cells, they would have the ability to produce further cells like themselves through self-renewal, and at the same time they could differentiate to provide various different cell types through differentiation, collectively leading to the formation of a tumor bulk. So I guess the million-dollar question is whether normal stem cells is the origin of cancer. Well, it has been estimated that about 3 to 6 genetic mutations are required to transform a normal cell into a cancer cell. And we all know that cancer develops from the accumulation of these genetic mutations. So, well, if this hypothesis holds true, that 3 to 6 genetic events are required to transform a normal cell into a cancer cell, then stem cells are probably, stem cells being the most long-lived among its progeny, are probably the only cells that would be long-lived enough to undergo the required number of mutations to initiate multi-stage carcinogenesis. So if we look at the properties of stem cells, you can see that stem cells already have the machinery for production of multiple generations of cells, even before undergoing any oncogenic genetic events. If you look at adult stem cells, they have several key features such as self-renewal, potential multilineage differentiation, as well as tissue regeneration. And when we study the properties of cancer stem cells, we found that cancer stem cells have similar properties to normal stem cells, but with the additional features that it would disturb the normal physiological processes such as angiogenesis, invasion, metastasis, and resistance to apoptosis. So this is the hypothesis put forward by researchers to explain the origin of cancer stem cells from normal stem cells. So in a normal stem cell, what you have here, with a normal stem cell would mainly divide symmetrically to give rise to another stem cell or a progenitor cell or a transit amplifying cell that would in turn divide into produce differentiated cells. With cancer stem cells, however, you would need to transform a normal cell into you would need genetic or epigenetic mutations or alterations to transform a normal cell into a cancer stem cell. And when the normal cell becomes cancer stem cell, what happens is that the cancer stem cell can divide both asymmetrically and symmetrically, giving rise to non-tumorigenic progenitor cells or transit amplifying cells, as well as potentially tumorigenic or even metastatic cancer stem cells. So the current paradigm for cancer stem cell definition, it's its ability to self-renew, that is to give rise to new stem cells, and its ability to reconstitute a given tumor tissue, and its tumorigenic ability upon enrichment. It would also be able to serially propagate when injected in vivo and undergo multilineage differentiation. And another important property, which would also be similar to normal stem cells, we all know that normal stem cells have intrinsic detoxifying capacity to get rid of toxins. And similarly, with cancer stem cells, it also has intrinsic detoxifying capacity that gives rise to its chemoresistance to conventional treatments. So in the remaining of the talk, I'm going to talk about cancer stem cells that have been discovered in HCC, colorectal liver metastasis, and also pancreatic cancer and some other biliary cancers. So to start with, development of cancer stem cells in HCC? Well, the stem cells in liver cancer, in the liver are still largely unknown, but it's well known that the hepatic stellate cells and also the oval cells are responsible for liver regeneration after hepatectomy or liver injury. So some studies have proposed that the hepatic stellate cells or oval cells are actually the origin of some of the cancer cells in HCC. So what happens is that when the liver is infected with, say, HPV, HCV, or chronic liver or chronic inflammation, mutations would arise. Mutations within cells, perhaps within cells of the liver, would transform the hepatic stellate cells or oval cells into cancer stem cells, leading to deregulated self-renewal, and also differentiation, collectively leading to the HCC tumor bulk. So this is a study performed by our group, and it's the first study to isolate cancer stem cells from human specimens of HCC. And in this study, what we did was we isolated the CD90 positive cancer stem cells from human HCC specimens and then injected into SCID mice to demonstrate its tumorigenicity. And interestingly, in addition to the CD90 positive cells, we also found a subset of cancer stem cells within the CD90 positive population. Namely, they're marked by an addition of CD44 positive cells. So these cells would be double positive for CD90 and CD44, and these cells these cells have the ability to develop lung metastasis in animal models. And here you can see that this is some table showing the rate of lung metastasis and different subsets of the CD90 positive cells. And you can see that in the CD90 positive, CD44 negative counterpart, the rate of lung metastasis is actually quite low, whereas in the double positive counterpart, you can see that there is a significant rate of lung metastasis formed in these animals. And so next what we did was we used a monoclonal antibody against CD44 in hope that it would reduce the growth and metastasis of these CD90 cells. And here, this is exactly what we see. We found that treatment with CD44 antibody could significantly reduce the rate of tumor formation and also reduce the rate of lung metastasis in these animals. And with this, we thought that CD44 monoclonal antibody may be a promising target, may be a promising target in HCC. And actually, there is a phase 1 trial ongoing using CD44 monoclonal antibody targeting HCC. And so next, we took this a step further. So thinking that if these cells were really tumorigenic and these cells were really tumorigenic, they could probably also be detected in the bloodstream. And we found that CD90 positive cells were detectable in 90% of blood samples from liver cancer patients, but none from the normal subjects or patients with cirrhosis. And we found a significant correlation between the number of tumor CD90 and circulating CD90 cells in the blood. And this study, we also showed the prognostic significance of CD90 cells in patients, showing that patients with higher CD90 in their blood would have a worse prognosis of in the disease. So next, since we were able to detect CD90, CD90 in the blood, we began to think that if this, the problem with detecting these cancer stem cells in the blood is that they're only present in very minute quantities, and that would make any subsequent analysis very difficult. So we thought that if we were able to detect it in the circulation, we might also be able to detect it in the pleural effusion of HCC patients. And since and pleural effusion of HCC patients, if we were able to detect these cancer stem cells, circulating cancer stem cells, would actually be able to provide a much larger source of these cells to be isolated and perform for further studies. So what we did was, this is a patient with lung and mediastinal and pleural metastasis. You can see the tumors here and here. And what we did was we tried to isolate these CD44 cells from the pleural effusion of this patient. And this is basically what we did. We isolated the pleural effusion of this patient, and then we performed a series of in vitro studies to confirm the stemness and to confirm the stemness of the cells isolated, and also make sure that it's of liver origin. We isolated the CD44 cells from these from the pleural effusion, and then we also did some in vitro studies to confirm its self-renewability and also its ability to differentiate. And what we show here is its ability to differentiate into the adipocyte lineage. And then what we did afterwards was we did some molecular profiling of these CD44 positive cells with different molecular markers. And what we did here was we isolated the CD44 positive and negative cells, and you can see here that there's a marked increase in the VEGF receptor level in the CD44 positive cancer stem cells as opposed to the CD44 negative counterparts. And so we treated this patient with a VEGF receptor inhibitor, namely pazopanib. So it's a pipeline compound by GSK. And here you can see this is the pretreatment CT of the patient. Again, you can see the tumor here and then the massive pleural effusion. 1 month after pazopanib treatment, you can see that the tumor was stabilized and also significantly increased in size after 1 month of pazopanib treatment. And more interestingly, you can see the pleural effusion almost completely disappeared. So, but unfortunately, this patient developed some complications with the use of pazopanib, so we had to stop. And you can see that 1 month after stopping pazopanib, you can see rapid tumor growth and massive pleural effusion again, which regenerated on both sides. And obviously, this patient subsequently succumbed to the disease. But this is just an illustration to show you how we could how we could isolate these cancer stem cells to personalize the treatment of with HCC patients. This is again another HCC patient, at which we isolated CD44 positive cancer stem cells from the pleural effusion, and then with this patient, you can see here that we identified CD44 positive cells. We identified mTOR to be upregulated, significantly upregulated, as compared with the CD44 negative counterpart. And so we treated this patient with which had multiple lesions at different segments of the liver. So we treated this patient with mTOR, RAD001, and then you can see that the largest lesion decreased from 7.5 to 3.5 centimeters with necrosis after onset of mTOR therapy. We also tried applying this. We also tried to apply this method to the ascites to isolate CD44 positive cells from ascites of HCC patients. And this is, again, another illustrative example showing you the isolation of CD44 positive cells from ascites. And again, here, we found upregulation of mTOR in the CD44 subset. So we gave our RAD001 therapy to this patient, and this is a CT obtained after 6 weeks after onset of RAD001 therapy, showing large areas of tumor necrosis. So, again, this is another, sorry. So that is basically just a very preliminary study to show how we could use cancer stem cell markers to guide treatment in HCC. And so the next question one would ask is, how is these cancer stem cells regulated? So MAATA, so it's also a group from our university, published a paper on the regulation of CSCs by microRNAs. So basically, microRNAs are small non-coding RNAs that regulate the expression at the post-transcriptional level. In this study, what Ma et al. Found that there was differential microRNA expression profiling of CD133, so it's another HCC CSC marker. So in this study, she found that there was differential microRNA expression profiling between 133 positive and negative cells, and she identified MIR130B as a regulator of CD133 positive cancer stem cells. And she performed further functional studies to show that the MIR130B, when transduced into 133 negative cells, would lead to enhanced tumorigenicity as well as greater potential for self-renewal and also greater resistance to chemotherapeutic agents. While to the contrary, when antagonizing the MIR130B in 133 positive cells, would lead to reduced tumorigenicity and chemoresistance. And it was also found that increased MIR130B paralleled its reduced downstream target, the TP53-INP1. So she showed that silencing this TP53-INP1 in CD133 cells could enhance both self-renewal and tumorigenicity in vivo. So apart from the CD90, CD44, and CD133 mentioned, recent study also has shown that CD24, another stem cell marker, has been shown to be upregulated in HCC. And in this particular study, also performed by our university, the study shows that CD24 positive cells were found to be very important in the maintenance, self-renewal, and differentiation and metastasis of tumors in HCC models. So what this study has shown is that inoculation of CD24 positive cells would induce tumorigenicity in SCID mice, and also they also showed that patients with higher CD24 positive cells showed much worse disease-free and overall survival. So I'm going to move on from HCC to colorectal liver metastasis. Well, colorectal liver metastasis, we know that about 50% of colorectal cancer patients develop synchronous or metachronous liver metastasis. Nevertheless, the presence of liver metastasis does not preclude curative treatment. In fact, about 20 to 30% of these patients are candidates for surgical resection, and following resection of the liver metastatic tumors, the 5-year survival can be as high as 30 to 40%. So with this in mind, we hope to search for specific markers that could be predictive of colorectal liver metastasis. And in fact, in colorectal cancer, the study of markers in normal intestinal stem cells and also colorectal cancer stem cells actually provides more clues to support that the origin of CSCs, and at least in colorectal cancer, seems to originate from normal intestinal stem cells because they share very similar markers. So we hypothesize that the metastatic capacity of these colorectal tumors may also be restricted to a subpopulation of cancer stem cells, but possibly with markers different from those found in non-metastatic tumors. So, and we think that colorectal cancer is a very good cancer model to study because it's one of the few cancers where both the primary and the metastatic tumor would be resected, and this actually provides us a unique opportunity to isolate and study the CSCs responsible for metastasis. So I'm not going to go into the detail as to how I identified this marker CD26 because this has already been published, but basically we identified CD26 as a marker for colorectal liver metastasis, as a CSC marker for colorectal liver metastasis. CD26 is basically a glycoprotein expressed on a variety of cell types, and it has been implicated in multiple biological functions, including tumorigenesis and tumor progression. So this is a very oversimplified summary of what we did. Basically, we have a fresh tumor tissue from a patient, either the liver. So this is the primary tumor, primary colorectal tumor. So we would have it dissociated, and we isolate the CD26 positive cells from patients, and we have the CD26 positive cancer stem cells inoculated into non-skid mice to allow for tumor formation. And then with the first generation of this xenograft formed, we would again dissociate the tumor bulk and then re-isolate and then isolate the CD26 positive subpopulation and repeat the process again and again. What we found was that as few as 1,000 CD26 positive cells can propagate and regrow into the tumor bulk, and that these tumors formed in serial passages were histologically similar to the parental tumor. So we further looked at these CD26 cells to investigate its metastatic properties. And again, here is what we did. We isolated the CD26 positive cells from the primary tumor. So these are from primary patient tumors, and implanted into the cecal wall of the colon. And then we observed tumor growth at about week 12. And we also tried to isolate the blood from the portal vein, and we were able to detect circulating CD26 positive epithelial cells during the growth of the metastatic tumor. And at about week 20, we see the formation of the metastatic tumor in the liver. We were able to repeat the isolation and implantation of these CD26 positive cells into the colon, and then we can see that the development of liver metastasis were of similar histology in successive passages. So we also looked at the invasive and migratory properties of these CD26 positive cancer stem cells in vitro, showing that these cells had enhanced invasion, migration, and also enhanced capacity in binding to ECM proteins. So next we looked at the chemoresistance of these cells. We all know that conventional chemotherapy targets all actively proliferating cells, and these are non-specific. However, cancer stem cells are actually more resistant to chemotherapy. So what happens is that with conventional chemotherapy, you would be killing the tumor bulk. You would be killing the tumor bulk, but leaving the CSC cells, which are highly chemoresistant, behind, and then the CSCs would subsequently regrow and into a more malignant tumor. So this is the chemotherapy which we've applied to in our animal models, showing a reduction in tumor size, but an enrichment of the CD26 population. You see here, so this is at day 14, where you see a significant reduction of tumor size, and at day 21. But at the same time, you see a significant enrichment of the CD26 population. And so this study actually identified the prognostic significance of CD26 cancer stem cells in the primary tumors as well as the metastatic tumors of CRC patients. So we took this study a step further to identify circulating cancer stem cells from colorectal liver metastasis patients. And I'm not going to go into the details here, but basically we try to compare the level of circulating CD26 cells as well as circulating tumor cells and in these patients. So we found that patients with CD26 positive cells in the tumor also had CD26 cells detected in their blood. And then we found that there was a correlation of CSC levels with Duke stage, showing that high preoperative blood CSC level, but not the circulating tumor cell or CEA, was associated with advanced Duke stage. And we were also able to establish the tumorigenic capacity of these circulating CD26 positive cancer stem cells. So when we looked at metastasis on follow-up, we found that a high preoperative circulating CD26 level, but not the CTC or the CEA, was able to predict the development of metastasis after resection of the primary tumor. And not only with the preoperative level, we also found a postoperative rise in circulating CD26 level, which preceded the clinical development of metastasis on follow-up of patients. So actually, I think this was supposed to read CTC and CA, so I think there was something wrong. So, yeah. So anyway, so this is supposed to read CSC. I think this is moved because I did this on my Mac, sorry. And this was supposed to read CTC, and then CEA goes here. So you can see that with there's a post-operative rise in circulating CD26 level, and which would precede the development of metastasis even on follow-up, whereas you won't be able to see the same pattern with CA or CTC. So what we found was that tumor CD26 and also serial monitoring of circulating CD26 seems in a post-operative period made predict the development of liver metastasis and may be a potential target for development of molecular targeting therapies to more effectively eradicate these cancer cells of the tumor. So I'm going to move on to pancreatic cancer. Pancreatic cancer is associated with a high rate of metastasis, high chemoresistance, and poor prognosis. Has been reported that a distinct population of CD133 cells was identified in pancreatic cancer. And this study by Herman et al. Also showed that a population of CD133 positive and CXCR4 positive cancer stem cells showed a metastatic phenotype using using pancreatic cancer cell lines, and they also showed that inhibition of CXCR4 with the drug could reduce tumor metastasis in mice. And more recently, this study published in Gastroenterology showed that using anti-CD44 antibody, they were able to inhibit pancreatic cancer. They were able to inhibit pancreatic cancer cell initiation and also post-radiation recurrence. So patients whose tumor expressed high levels of CD44 had a median survival of 10 months, compared with less than 43 months for those with low levels. And anti-CD44 inhibited growth and metastasis was able to significantly inhibit the growth, metastasis, and also recurrence of pancreatic cancer in the mice model. So cancer stem cells in biliary cancers has not been very well studied, but there has been scattered publications of, say, for example, there's one study on cholangiocarcinoma showing identifying CD24 and CD44 positive cells as cancer stem cells in cholangioCA. And also CD44 positive and CD133 double positive cells seem to be important in human gallbladder cancer. So, well, these are the only publications I was able to find. So I guess the therapeutic implications of CSCs in these biliary cancers remain to be elucidated. So to conclude, cancer stem cells play a critical role in the growth and metastasis of HPB cancers, and they contribute to generally high chemoresistance and poor prognosis of HPB cancers. So evaluation of the tumor as well as the circulating cancers, which I've showed you, circulating cancer cells, which I've showed you, in these HPB cancers may provide prognostic value and therapeutic guidance on the adjuvant therapy after surgical resection. And it would also aid in the elucidation of molecular pathways of which would help to develop new therapeutic strategies to target these tumorigenic subpopulations. So thank you.